All right. So welcome back. So this is a little bit of an overview of the practical, and also to announce that some part of the practical will be focusing on a little bit of we'll give you a little bit of lecture aspect, although it will be mostly exercises, but there will be some theoretical concept there. And so the practical is going to be mostly focused on just trying to explore through simulations and changes in allele frequencies and genotype frequencies. And so I've alluded to that in the second part of the lecture that some of the what I refer to here as evolutionary forces, and so that language is partly coming from the fact that the population genetics theory literature borrowed a little bit from the physics literature. And so you have terminologies like evolutionary forces, but think about it as drivers of changes in allele frequencies. And some of those evolutionary forces include things like mutations and migrations, which I've talked about in part two, natural selection, which in the interest of time, we're not going to cover in this lecture. But other forces like genetic drift, or demography here, essentially, things like contraction in population size or expansion in population size, which can happen for various reasons. Those will have an impact on genotypes frequencies, and this is something we will explore. But what I'm going to cover now is another part that we won't have time to cover during the practical, which is the right fixation index, or FST. So this is a very popular statistic you will see in population genetics theory, and it is a statistic that is aiming at measuring what is called genetic differentiation. And so it's something that is applied to now not individuals, but populations. And we have one population here, say another population here, and we want to know how genetically similar or dissimilar those populations are. And so what I'm illustrating now is just one potential way of understanding why population can end up being genetically different. And that is because of this evolutionary force called genetic drift. So in a nutshell, genetic drift is a very simple force in that is you have essentially just a random sampling of who gets to have children for the next generation. Imagine that we all in some sort of big room, and a certain number that is predetermined will is selected at random to leave the room and have children for the next generation. So this is a sort of conceptually what genetic drift is. So there is no notion of fitness here. So it's an evolutionary force that is neutral in that regard. So what determines whether people have children and contribute children to the next generation is completely random, right? And so under this evolutionary force, we can show that allele frequencies will change and fluctuate, and this is something we will explore at length during the practical. But here, assuming that we started from this original population, which I call the ancestral population, and then we essentially have a subdivision, right? The people part ways, and we sort of define different groups. We have group one, group two, and group K all the way. And if we wait long enough, and within each of those groups, mating happens randomly, that will produce this phenomenon called differentiation, where allele frequencies will slightly differ, and the more we will wait, they will differ more and more. And so coming back to FST, which I'll talk about in the next few slides, it's a way for us to measure the degree to which that differentiation has happened. So what I'm describing is a very simple evolutionary force where we have a structure in the population. And so here I'm also taking this opportunity to define what a population structure means. So population structure will mean that within our population, there will exist subgroups that are genetically differentiated. right? So if I go back to that particular example where we had our ancestral population and all the daughter populations, if now I'm defining somehow I'm defining a population that has individuals from all the daughter populations, well, I would have created a set that is slightly heterogeneous suddenly because I know that because of genetic drift, individuals in the daughter population one will be a little bit different from at least the frequencies of alleles will be a little bit different in individuals from population one and derived population two all the way to population K. And now, if I suddenly aggregate everybody without knowing, there will be some structure in the population in that some individuals will be more genetically similar than others, right? And that has as happens even if we don't have related individuals, and that kind of a setting is referred to as population structure. And what I'm describing here is a broad approximation. I think the demographic history between population, human populations is a little bit more complicated than that because you have migrations, you have bottlenecks, etc. But as a first order approximation, we can approximate it with a tree structure like this, where we have an ancestral population, which most likely originated from Africa, which had led to daughter populations, including contemporary African populations, but also populations from pretty much everywhere on the planet, including European and East Asian ancestries depicted in this schematic from Martin et al. 2019. So now let's go back to defining what is this fixation index or FST. So I'm going to take the definition proposed as right as a ratio of variance. So now thinking about our those going back to individuals. So I know this is a statistic that compares populations, but imagine that those populations will be composed of individuals. And so in in a population, individuals will have what I would call an allelic state. So if you think about one particular allele, you can have the allele or you don't have the allele, right? So you have this is allelic state, and this is random, right? And that random variable will have a different distribution within each of those populations, right? Because of genetic drift, as said, we would expect the frequencies of those alleles, of any allele actually, to be slightly different because of drift, but also because of any evolutionary force that will create those differences. And so the FST statistic is a measure that differentiation. So it's something you can do, you can apply to pairs of populations, but you can also calculate FST across an entire set of populations. So it's not just a pairwise statistic, although most of the time it is utilized as a pairwise statistic, but you can have FST across, say, 5, 7, 10 populations if you want. And so the way it is defined as the variance of that allelic state uh, between populations divided by the total variance of that allelic state, which will include the variance between population and the variance within population, which here you can think about easily as if you have now going back to individuals in each of those populations, well, you can actually observe their allelic states and calculate that variance of that allelic state, and that will give you that quantity. So, which means that you can also recalculate FST as 1 minus the variance within divided by the total variance, which is another way of defining this. So a few observations. What is an FST of 0? FST of 0 means that there are actually no meaningful frequency differences between populations, and which would also be evidence of the absence of population structure. So you're looking at your total set of populations, or at least of those groups of individuals. And that total set, essentially, you don't have don't have some groups in that population, in that total set, sorry, that are more genetically similar than others, right? So you have this pan-mixia in a way that almost as if everyone could mate with anyone. The opposite, the other extreme is when FST equals 1, which means that you have a maximum differentiation. And maximum differentiation mean that all the genetic variants that exist is actually between populations, right? And so that typically happens when you have a particular variant that is fixed in one population. Uh, let's say in one population, everybody has the A allele, and in the other population, everybody else has the B allele, and then you don't have that. No, you have no heterozygotes in those cases. So that's the definition of FST. Now let's talk about how do you estimate it. And this is what is interesting. So they are again different. I guess here I focus on one definition, but there are actually many estimators of FST. We have the classical Weir Cockrum FST, which has some formula. We have the Ney FST and the Hudson FST estimator. So those are the names of the different estimators that exist. Um, I'd like to point out this very interesting and important paper by Bhatia and Patterson, Sankaraman and Price, which gives a very nice overview about the definition and interpretation of FST. There is a bit of a focus on rare variants, but even if you're not interested in rare variants, I will highly recommend reading that paper. And that paper highlights another definition of FST, which is related to again variance, but it's a variance of allele frequencies. So you again having this notion that you have derived populations and allele frequencies, and now is random. So what I introduced is the randomness was around the people who gets to be in the population and their allelic states. But here, the randomness is about the population itself, right? So you have allele frequencies becomes a random quantity, which here on its expectation using this particular model, the Weir and Hill model, is the same as in the ancestral population across all derived populations. However, you have fluctuations. The allele frequency will vary around that of the ancestral population, and the degree to which it will vary will be a function of FST. And that's also another definition proposed here by Bhatia. And this one is a very good one if you are interested in simulating allele frequencies directly without simulating individuals and then derive from those simulated genotypes deriving allele frequencies again. So that gives you a very powerful framework to simulate allele frequencies using FST and some predefined set of differentiation. Just to close on this, just a couple of things we can highlight is that the FST varies across the genome. So what I presented earlier was the particular locus, but you can repeat the same thing across the genome. And interestingly, it's not because you have a strong differentiation in one particular part of the genome that you should necessarily expect a strong differentiation across the entire genome. And this is empirical data showed in the Laird and colleague paper, which compares allele frequencies between the European ancestry populations, the CU subgroup in the 1,000 genome data sets, which is in the publicly available data set, with population from East Asia from the CHB sample from that same data set. And the bottom is another comparison. It's the comparison between that CU data set with West African Yoruba population from 1,000 genome as well. And so what we see, x-axis is not labeled, but represent genomic positions of those loci, and the y-axis represent FST. And as you can see, there is quite a bit of variation. And they highlighted in this paper part of the genome where FST was quite large, and in particular around this enzyme that is involved in alcohol metabolism, which is quite differentiated between European and East Asian populations. But just to give you a notion of what is the range that you would expect for FST. So typically when you compare populations, in particular a population that are on different continents. And then here the geographical element is important because it's continent here is a proxy for isolation. These populations have lived apart from each other for sufficiently long, and sometimes that is because of natural barriers like oceans or mountains. But those sort of you expect for continental groups to have FSTs between zero point 0.1 and zero point 0.2. Typically within continents where you have a lot more migration happening, and if you say you compare populations from Central Europe and Southern Europe, you would have FST between 1 and 5%. And typically, when you look at subgroups within a country, so again, very much smaller geographical spaces, meaning that there are more opportunities for people to migrate and to have a bit more mating happening between different parts of that country. That usually leads to FST values that are lower than 1%. So that gives you a notion of what to expect. Although, again, being said, if within a country you can have subgroups that are highly differentiated, if historically they haven't been in contact, partly they've been separated by a mountain, and it was tricky to travel through that mountain. That could also lead to very strong differentiations between groups that have lived within relatively small geographical areas. All right, so that was this part, which, just to summarize, gave an overview about what the practical is going to be about. Is essentially going to explore through simulations how allele frequencies and genotypes frequencies change using playing with different assumptions. And also took that opportunity to introduce this fixation index parameter, or FST, which has different definitions. I introduced an individual level based definition using allelic stages, allelic states, or another definition, the Weir-Hill definition, which directly built it from the allele frequencies, and also highlighted different estimators, some of which we're going to use during the practical. So I'm stopping here, and then we're moving to the last part of the practical of the lecture, which is going to be about linkage disequilibrium.